The exam will be 34 questions. There will also be 2 bonus questions, okay? You're going to have your full 50 minutes to do the exam unless you have any accommodations. And the accommodations will extend the time that you have. So you can stick around afterward and complete your exam. I know some folks have set up for separate proctoring through the testing center, so that may be available to you as appropriate on a individual basis. So that would be the proctoring. You have to have your cameras on. If you have a non-functioning camera or you're having technical trouble or you're in an area that's not well lit or anything like that, please let me know before the test that you're going to need some special exception because of your technology issues or computer or whatnot. Just let me know before. You can send me an email up to Sunday evening if you have any of those issues. So you have to be live on Zoom and you have to have your cameras on. Yeah, so that will be the format for the exam. And if you have any questions on that, please let me know. You can drop them in the chat and we can go over that. I will also create a breakout room for questions. So if you have questions on any of the items or any of the wording or images or anything like that, just send me a direct message during the test and I will pop you into a breakout room and we can jump on and kind of go over your individual questions. See, am I forgetting anything? No, I think that's it. But if there are any other questions, feel free to let me know anytime during this review. All right, so let's begin to think about some of the content that we're reviewing here for this first exam. And we're going to start right at the beginning where we talked about the fluid compartments, homeostasis, and the simplified body plan. So we want to think about our fluid compartments. Good question. Do you need scratch paper? You can have scratch paper, and I typically ask everyone to hold up their scratch paper at the beginning. So if you have that, definitely have it handy at the start before we're ready to go, and just make sure it's completely blank on both sides. Okay, good question. So our fluid compartments we spoke about, there were 2 major ones. So we have our total body water, which is all the water in the body. And then we can further divide that into the intracellular fluid, which is the water that is inside cells. We said this was the majority, right? About 70% of total body water versus the extracellular fluid, which is all the water outside cells. And we said that that was comparatively about 30%. We can also further divide the extracellular fluid into 2 smaller compartments. So we have the plasma, which is about 10% or so of that remaining 30%. And then we have the interstitial fluid, which is about another 20%. And just to further describe these 2 compartments, the plasma is going to be the fluid inside blood vessels. The interstitial fluid is going to be fluid around tissues. Okay. So these are general body compartments and the relative proportions, right? And again, I don't need you to memorize these numbers, but know that most of the water in the total body water is inside cells. Most of the extracellular fluid is going to be in the tissues. Okay. We also want to think in this chapter about homeostasis. Which was the second major part of chapter 1. And the pearls or the important aspects of chapter 1 and homeostasis are going to be the negative and positive feedback. So just to describe these two mechanisms. Negative feedback, we describe that as being changes that correct the error signal. All right. In other words, it's going to move our regulated variable towards set point, back towards set point. So moves regulated variables, which could be a lot of things, right? Body temperature, pH, blood pressure, all these things back towards set point, okay? And a really important hallmark of homeostasis is that it's not perfectly done. It's always a continual monitoring and adjusting, and it's really striving to maintain this set point. Let's shift and think about positive feedback. Positive feedback. And positive feedback, first of all, we said it's extremely rare. So really important to know that it's not as abundant and as important as negative feedback. We also said that it's a brief period of time. It's not going to be indefinite and continuous like negative feedback, but it is a brief period of time. Oops. So a brief period of moving, again, our regulated variables away from set point, okay, which was for a desired result. And then ultimately, positive feedback would end with a negative feedback event. So it's going to end with a negative feedback. A negative feedback event. Okay. Let's list a few specific examples because it's so rare. We can think about some very specific examples that highlight it. Some of the examples would be, we talked about blood clotting. So the clotting of blood is an example here, as well as the strength of uterine contractions during childbirth. Okay. And so these are two examples where for a short period of time, the variables, which in this case would be the blood, right? The bleeding that's happening at the site of injury. And in this case would be the strength of uterine contractions. Those things move away from what the body would normally want and as far as homeostasis. But it's for a short period of time, which is for a desired result. In this case, it's for the birth, right? The birth of the fetus. And in this case, it's for allowing clotting factors to get to the site of injury. And then ultimately, both of these cycles would end with a negative feedback that would stop the bleeding in this example and would cease the contractions of the uterine muscle in the second example. Okay. So those are kind of the main points to sort of focus your studying around for chapter 1. Know your fluid compartments, know the proportions, know which one is where, and then also know the mechanisms by which we maintain homeostasis and be able to compare and differentiate negative and positive feedback. Okay. I'll just take a brief pause to see if there are any specific questions, and then we'll move on to our second topic. All right. Let's move on to think about biomolecules and enzymes. So this was packet 2. So we're thinking about biomolecules here. Okay. First off, we said that these are the major building blocks for essentially our the major molecules that are the building blocks of our body, right? So the major building blocks of our body. We also said that the major elements that form these biomolecules are going to be carbon, excuse me, hydrogen, nitrogen, and oxygen. So various proportions of these three elements are going to come together and form our carbohydrates, proteins, lipids, and fats. So let's kind of describe the difference between some of these elements. So lipids, and then we can think about carbohydrates, proteins, and nucleic acids. Okay. Now we can classify lipids differently than the other three because lipids are sort of unique. They are non-polar, okay, which means that they do not interact nicely with water. Okay. They are also going to form examples important such as our phospholipids. So some of the examples would be triglycerides, phospholipids, which are extremely important for the phospholipid bilayer, which is our cell membrane. And I'm also going to put amphipathic here. That's extremely important feature of phospholipids, amphipathic, which means that they have an area that plays well with water and an area that does not. So a polar head and a non-polar tail, okay? There were other examples that we talked about, cholesterol, etc., as well, right? So forming our hormones and so forth. So definitely know the lipids, know their unique features and that they're non-polar, and know some of the examples of where we find them, and the different subtypes as well. Now we can classify the other three biomolecules as all being polar. So these are going to be more polar, and they are going to exist well with water, right? So I'm just going to write polar for all the others. Some additional features of carbohydrates is that we can think about monosaccharides, disaccharides, and polysaccharides, okay? So definitely know the monosaccharides versus disaccharides versus your more complex polysaccharides, okay? And know how the binding of that differs. So know the difference between a condensation which is where we remove water in order to form a bond. Okay, or a hydrolysis reaction, which is where we insert water to break that bond. Okay. So know again, those mechanisms of making more complex monosaccharides and all the examples. So definitely know the difference between, I'm going to put in brackets here, glucose. What is glucose, right? It's a monosaccharide versus fructose, a disaccharide versus glycogen, more complex, okay? Proteins, similarly, so know the primary structure, which is really the peptide bonds. Put that in brackets here, peptide bonds, versus secondary versus tertiary and even quaternary structure. Okay. And then for nucleic acids, we talked about several examples. So just know the examples of these. So for example, cyclic AMP, we talked about purines versus pyrimidines. Okay. We talked about NADH, which are going to be our carriers, and FAD as well. Okay. So think about some of the examples for these biomolecules. You want to know the specific subtypes and just examples, okay, of that. All right, let's go ahead and move on. I'm going to take a quick pause again. Let's think about enzymes. And so enzymes basically decrease the activation energy of a reaction. In other words, they speed up the reaction. Not speed up, excuse me, they make the reaction more favorable or more likely, right? So they make the reaction more favorable, right, or more likely. They also, if we think about some of the examples of factors that increase the rate of enzymatic activity. So factors that would increase the rate of enzymatic activity those would include the individual catalytic weight of each enzyme. So each enzyme differs in its individual catalytic weight. So the catalytic weight is one factor. The substrate and enzyme concentration. So how much substrate or enzyme is present is another factor. So the substrate and enzyme concentration. We can also think about the temperature and pH. Okay, these two things are going to increase the enzymatic activity to a certain point, but beyond that point, remember, these are proteins, and beyond a certain temperature and pH threshold, it's actually going to denature those proteins. So that's going to certainly affect the rate of enzymatic activity to a point. And then the affinity of the enzyme for its substrate. Okay, how likely is the enzyme, or how attracted is the enzyme to the substrate? Another important aspect here is thinking about different types of reactions. So we spoke about the endergonic. Another way to express this is endothermic reactions, and these require energy. Another term we can use here is anabolic, right? And an example of this would be making proteins from amino acids. So using proteins and building those up to form amino acids. So making proteins from amino acids. And we can compare that to exergonic reactions, which are also exothermic. And these are going to donate or release energy. You can also refer to this as being catabolic. And the example here is to break peptide bonds to make amino acids. Okay, so just be able to differentiate these two types of reactions and some examples, right? How are we using energy to store within the building up of these biomolecules, for example, amino acids to make proteins, or are we breaking those bonds and releasing that energy and breaking those smaller components of the biomolecules? Okay, so two examples as well. Okay, let's keep going. Let's move on to cellular respiration. So cellular respiration. Okay. Let's think about cellular respiration. So essentially, to sort of summarize cellular respiration, is it's harnessing energy from the biomolecules in the food that we eat, right? We bring in these biomolecules in our diet, and we need to liberate the energy that's stored within these molecules and transfer that into a source that our cells can use, which happens to be ATP. So we harness the energy from biomolecules to synthesize ATP, really important. And this ATP is to do cellular work. Okay. So that's kind of a description of cellular respiration. Let's think about the processes involved. So the processes involved, first off, is glycolysis. And we're going to talk about the major products of each of these steps and some of the important features. Important about glycolysis is that it occurs in the cytosol. All right. Its overall product yield will be 2 net ATP. It's also going to generate NADH. So again, electrons being shuttled to the mitochondria. And then the final product here will be pyruvate. Actually, 2 pyruvate per glucose. So 2 net ATP, NADH, and then 2 pyruvate. And the purpose of glycolysis is to take glucose. So we start with glucose and then we end up with these products. Electrons going to help with the electron transport chain. Pyruvate going to the linking step, which is the next step that we'll talk about. And then ATP being a part of the overall net ATP made per molecule of glucose. Let's think about the shorter intermediate linking step. So the linking step is an intermediary step where we take this pyruvate and we generate acetyl-CoA. This happens in the mitochondria. Okay, that's the site of the linking step. And we're going to insert pyruvate. So our products here, our substrates here will be our products from glycolysis. And we're going to yield acetyl-CoA. We're also going to yield some carbon dioxide as a byproduct, but we yield no ATP and we yield no electrons, right? It's really an intermediate step that converts pyruvate into acetyl-CoA. The third more important and meaningful step is the Krebs cycle, or also called this the TCA cycle or citric acid cycle. This also occurs in the mitochondria. And this is going to yield us 2 ATP as well. It's also going to yield us some more electrons, but this time we have 2 of our carriers engaged. So NADH carrying electrons, and then FAD as well. And then we also yield some carbon dioxide as a byproduct. Lastly, the most important step here being the electron transport chain. This occurs across the inner mitochondrial membrane. All right, this is going to yield us much more ATP, so much more significant. This will give 34 ATP, okay? And it's going to do so by way of oxidative phosphorylation. Which means that this is aerobic, so oxidative phosphorylation, which means that oxygen is required as the final electron acceptor. So oxygen will be standing by to be the final electron acceptor. Okay, very important, this last bit here. Okay. So that is these are the stages of glycolysis. I also just want to reiterate that the first 2 steps here, so this step and this step, are going to both make their ATP, so this smaller amount. This is going to be made by substrate level phosphorylation. So substrate level phosphorylation is going to make 4 out of the 38 total ATP, whereas the electron transport chain by way of oxidative phosphorylation is going to make 34 out of the 38 ATP, right? So the bulk of the ATP will be made from the electron transport chain.